my name is Taylor Haight, and today I'm going to be presenting my honors project on Woodland and Vernal Pool salamanders. So as a bit of background information, a vernal pool is a type of wetland habitat with no permanent inlet or outlet. That means that in the springtime, they get filled up with water from melting snow and rain, and they usually dry up by the summer. Like other types of wetland habitat, they provide many important ecosystem services, including flood and erosion control, water purification, and carbon sequestration, and habitat to many species of birds, reptiles, invertebrates, and amphibians, including salamanders. Salamanders are a very important part of our forest ecosystems. They serve as both predators and prey in the food chain, and they're also an indicator species because of their sensitivity to environmental changes. So by measuring the number of salamanders you have in a forest, you can get an idea of its overall health. By feeding on organisms which break down organic matter, they also help mitigate the effects of climate change by preventing those populations from becoming too large and breaking down that matter quickly and releasing large amounts of carbon into the atmosphere. These are a few of the species of salamanders we have around campus. One of them is the Redback Salamander, which is the most abundant forest vertebrate in our area. We also found a four-toed salamander, like shown in this picture. These are very rarely seen and have very specific habitat requirements. They're a data deficient species. We don't know a whole lot about their populations, only that they're believed to be declining in this area. So it was really cool to be able to find one here. The experiment we set up was set to answer the question, why don't these salamanders we're finding around campus seem to be using the vernal pools at RTPI? So to do that, we set up surveys at 3 vernal pools, one at JCC and 2 at RTPI. We set up 50 by 50 meter quadrats around these pools, and we were doing surveys to measure the number of species found in each of these quadrats, their size, the location, and the type of log they were found under. So the type of log refers to the hardness or the freshness of the log. So as we're turning them over to check for salamanders, we were just measuring on a scale of 1 to 3 how decayed they were, how easily they were falling apart. This is the survey areas at JCC. So the vernal pool was found in plot B in this corner here. And if you were to turn this around vertically, plot A would be closest to College Park and plot D would be closest to campus. These are the survey areas at RTPI. So the first vernal pool is found right beside the road and the parking lot. The second is found in the woods behind the building. This is a summary of the results from JCC. So as you might expect, the majority of salamanders are found were red-backed, and most of them were found under hardness 1 logs, so those freshest logs. Plot C actually had the greatest number of salamanders and the greatest number of species, which is a little bit different than what we might have expected because plot B actually is the one that contains that vernal pool. The results were very different at RTPI. We didn't find any salamanders in either plot for any of the 3 surveys we did there. The only salamander we ever found on that part of campus was a redback salamander 23 meters away from the second plot. So the next question was why? Why is there such a big difference between the abundance of salamanders at JCC versus RTPI? And I think it's a little bit easier to answer that question for plot 1 at RTPI. Because even though there was dense vegetation and plenty of available cover, which would normally be very good for salamanders, it was also very close to that paved parking lot and the road, and the trees there were mostly white pine and deciduous trees, which is different from the plot at GCC and the second plot in the woods for RTPI. So all of these factors contribute to more evaporation of water. So in the springtime, the pools might have been drying up too quickly for larvae and eggs to mature. And it also means that for the rest of the year, like when we were doing these surveys, it's easier for the soil to dry out, which is not good for salamanders. It means that they're more vulnerable to drying out themselves. We also know that the litter was made up of mostly needles, and we know that terrestrial salamanders tend to prefer deciduous leaves to needles, which also might be why we weren't finding them there. There could have also been a difference in acidity due to those needles, but because we couldn't test the pH of the soil, we don't know that for sure. It's a little bit more difficult to answer the question for the second plot at RTPI because it's so similar to the plot containing the vernal pool at JCC. Both of them had a mix of hemlock and deciduous trees, Both of them had plenty of available logs and leaf litter and similar invertebrates, so a food source probably wasn't an issue. And neither site had a significant number of worms, which, if they did, might have meant there was competition between salamanders and worms, because worms actually break down the leaf litter that salamanders need. So if there's a lot of worms, then it creates an environment that isn't suitable for them anymore. But one of the potential factors might have been the soil itself. Vernal pool soil is poorly drained, which is good in the springtime because that's what allows it to fill up and create that habitat which is necessary for breeding. But for the rest of the year, it's too sticky for burrowing salamanders. They can actually get stuck and drown in the soil, so they would tend to avoid that when it's not filled up. And because the plotted RTPI was made up of mostly pool, and we weren't looking at the surrounding area like we were at JCC, that might explain the difference in the abundance of salamanders. And it might also explain why we found that one redback salamander far away from that pool itself. Another potential factor might be the type of forest. So we know that most salamanders prefer mature and interior forest, and the plot at the woodlot at JCC is not only older, but there's less habitat fragmentation in that area. So if you look at the plot at RTPI, not only is the forest younger, but the plot we were looking at is very close to a road and a path in the woods. So that might be why they were avoiding that area versus going further into the woods or choosing the plots at JCC rather than RTPI. Another factor might have been contamination from the nearby road. So if any salt from the road or other contaminants were to get into the soil, they could very easily toxify it for salamanders since, again, they are so sensitive to changes in their environment. And this is just showing you that difference between RTPI and JCC. So if you look, the road at RTPI is very close to that plot we were looking at, and there's also a cleared path running through on this side of the plot. Versus at JCC, where there's a lot more open area and undisturbed habitat. It would be very interesting to see this experiment repeated when the pools are filled up to see if any of the trends still exist. Thank you for listening.